Yeah, and so in this talk, I'll try to wrestle a little bit with what we're learning. I'll talk about this neural signal propagation atlas that we've been measuring, that we measure in the worm. The worm's shown here, right? And those are some neurons in its head. It has 302 neurons. There's about 200 in its head. And I'll talk about kind of what we're making sense of our measurements with respect to the connectome. So for much of the Simons Foundation period, we focused on studying how patterns of neural activity generate actions or represent information from the environment. We've done this in freely moving animals to study neural dynamics of underlying locomotion, or we've studied neural dynamics of decision making. And over the years, we've gotten more and more interested to try to understand, well, in addition to performing computations and generating actions, is it very loud? Move it down. Okay, good. It's just how I hear it. In addition to trying to understand how those dynamics operate along this pathway, we'd like to know, what is it about the network that gives rise to the dynamics we see? And in particular, when I think about properties of the network, one of the most salient properties is the connectome. And so in the second half of the talk, we'll try to ask, how is it that we go from a static connectome to getting patterns of activity and dynamics. And in the worm, like in other organisms, this connectome comes from electron microscopy. Okay, and so the first step to try to ask this question is we wanted to measure very systematically how signals causally propagate through the network. So this is the team that did the first set of measurements I'll share with you. And so the idea was we wanted to measure the entire network's response to single neuron optogenetic activation. And for lack of a better term, we call this signal propagation. The paradigm here is we're going to go one at a time. We'll stimulate each neuron, so stimulate neuron A, and we're going to measure the response in A, in B, and in all the other neurons. And that's going to give us information about the strength, the sign, the causal arrow we get for free, and the temporal properties of the network's response to stimulation. And there's been a lot of great work in this area, including from many of you in the room. And what we're trying to bring to this is by working in a smaller organism and with some technological advances, we're trying to do this. What's new is to do it at brain scale and cellular resolution. And we want to contrast this to an approach that we and many others have taken in the past, which is to look at correlations in spontaneous activity. If you have a correlation matrix, it's by definition symmetric. You get symmetric couplings. It's much harder to tease out information about causality. It doesn't directly capture the temporal profile of the response to a neuron's activity. And I think the last one resonates most with me, which is that you're limited to neurons that are spontaneously active. So if you have a correlation matrix and there's a neuron that never does anything, you don't have any information about it. But in a perturbative regime, you can go in and ping that neuron and see how the network responds. So there was a lot of technological development we had to do these measurements. I'm going to mostly skip over it. I'm just going to say we're doing 2-photon stimulation. We're doing spinning disk confocal for whole brain calcium activity because we want to measure all the neurons while we stimulate. There were some challenges to get healthy animals where every single neuron is expressing both an optogenetic protein and a calcium indicator. We had some tricks for that. Another really important trick is we're using the NeuroPal system to identify all our neurons so that we can map the recordings we get from each neuron onto the connectome and across animals. And so in this system, you use not only the position of each neuron, which is stereotyped, but you also get this color-coded gene expression profile that allows you to uniquely identify each neuron with its name. And so here's what a recording looks like. So unlike most of the work in the lab that we do in freely moving animals, this one is an immobilized worm, so the worm goes here. And then this is a Z projection of a sped up recording where we're doing calcium imaging. And every 30 seconds, we're going to stimulate a neuron. So there we stimulate that neuron. You see that neuron respond, and some others respond. And then we'll move over. We'll stimulate a different neuron, and you'll see that neuron respond, and other neurons in the network respond. And we do this over and over again. And so this is what a single trial recording looks like. So each trace is a different neuron measured simultaneously. The identities of the neurons are named here on the left. We record for about 40 minutes. And on top, you have information wherever there's this gray line is where we stimulate a different neuron. So I'll walk you through this. For example, here we stimulate neuron I3. you can see there's many neurons that respond. Among them is I3. We record from I3, and it responds as we would hope. I'll point your attention to one example. This is neuron M3L. It also responds. Because we have the connectome, of course, we can look and we can see that there's many monosynaptic and polysynaptic paths through the connectome for signals to potentially travel from I3 to M3L. And it's worth noting that when we look at M3L's response, we're seeing the cumulative sum of contributions from all of those paths. And then we can repeat this across animals or across different trials. So here I'm showing paired recordings. So you can see I3's response to stimulus and M3's response. And one of the ways we can represent this is we can take an average, so we can look at the average response. And we did this over and over again, and we generated what we're calling an atlas. And one way to represent that is in this matrix. And so the way you read this matrix is here, these are the neurons you stimulate, and this is the neurons that respond. And so that I3 to M3L is just one pixel here. We're really proud to have done this for a very large number of animals to try to get a lot of coverage. So we've done 113 animals, 9,000 stimulations covering 2/3 of all possible neuron pairs. It's over 6 terabytes of imaging data, and thanks to the Simons Foundation, we got it all in NWB and up on Dandy, and we're trying to make these resources really broadly available. I want to say that, just having this atlas is already very valuable in its own right for the worm community. Each one of these neurons tells a story. So for example, I can point to one neuron pair that I hadn't heard much about, AVJ. When you stimulate it, AVD responds. If you go to the literature, based on the connectome, the AVJ had been thought to be involved in egg laying and forward locomotion. But now you can immediately update this hypothesis because if you're a wormy person, AVD is well known to be involved with backward locomotion. So you say, okay, new hypothesis, maybe this is involved with backward locomotion. And we're not content just to look at the average amplitude of the response. We like to think we get a lot more temporal information if we look at if we define something like a transfer function here. So for each pair recording where we have a neuron that's stimulated and its response, we can define a kernel such that if you convolve that kernel with the upstream neuron's activity, it gives you the downstream neuron's activity. And this is actually a pretty useful way of making very reasonable predictions about how the network responds. And so we have a little simulator here that just allows you to draw in arbitrary waveforms to inject into the network, and then you pass it through this convolution, and it does a good job actually predicting how the network responds, and it actually matches pretty well what we see if you drive the network and look at its spontaneous activity, for example. Okay, so and then we're excited to expand not just an atlas of the hermaphrodite, but we want to look at other brains that are very similar but have known and defined differences. And in this case, that's called a male. So male brains have a few more neurons, slightly different wiring, and a known single-cell transcriptome that differs. And so we're excited to be looking at how those differences influence signaling. So for the rest of the talk, so that's all about measuring. For the rest of the talk, I'm going to talk about how we're wrestling with how to make sense of what we measure with regards to the connectome. And as Larry and Mala had said, we're in this new age of the connectome. And so a reasonable question is, how does the signal that we measure relate to the wiring? And so here's a preview of what we've learned so far, and I'll show you the evidence for it in a second. But the first thing is, what we found is it is still, I would say, challenging to predict signaling de novo from a connectome alone. And so we'll talk about that. At the same time, it seems that given your measurements of neural activity, the connectome is uniquely informative for trying to make sense of what you see. And those aren't as in tension, I think, as it might seem. And I'll show you evidence that we see extrasynaptic signaling, including on fast timescales, that isn't present in the connectome. And so we'll work through this all together. OK, so what's the first thing we wanted to check with respect to the connectome? Well, you should certainly hope that neurons that are directly monosynaptically connected, that they should be more likely to evoke responses than neurons that are further, that have a longer minimum path length through the network. And that's exactly what we see. So if one neuron is monosynaptically connected to another, it's much more likely to evoke a response if you stimulate that neuron than the further away you get. Okay, but one of the things that had been challenging, one might hope that if you gave me a connectome and information about the number of synaptic contacts and its transcriptome, so you knew something about what's excitatory or inhibitory, you might be able to just predict our signaling de novo, and that's something that we haven't been able to do. And maybe, depending on your prior, maybe that's not surprising because you need information about timescales, reversal potential, sigmoids, and conductances, and those are molecular details. But here's one example where we took kind of an off-the-shelf established model. There's a kind of mature modeling field in C. elegans. And we, in silico, repeated our stimulation experiments. And if everything agreed, it should be on this dashed line. But it doesn't all fall on the dashed line. We have a negative R squared. One way you can think about this is, of the high confident responding neuron pairs that we measured, only about 20% were predicted to have large responses in the model. So we would say that it's hard to predict de novo. Okay, but maybe you'd say, okay, well, forget predicting the individual responses, but the network science has a whole field that focuses on kind of this implicit assumption that properties of the network, like the architecture, are informative and helpful. So we wanted to ask, okay, so maybe if we looked at the community structure, that should be more similar. And so we used a stochastic block model. It's a kind of standard network sciences approach to break up your network into communities. And there, I think the story from these network science approaches is that really, really striking network features seem to be conserved in both the connectome and the signaling networks, but other things do not. And so one of the most striking things that comes out of the connectome is the subnetwork of the pharynx. There's a bunch of neurons that are pretty isolated from the entire rest of the network that jump straight out. When you do stochastic block modeling and break it into community structure, that's also the case in the signaling network. You see that the pharynx and the signaling network also pops out. And you can see this in this matrix of numbers of neurons that overlap in these different communities. But most of the other communities don't map one-to-one or are not neatly contained from one into the other. And this seems to be the theme. I won't tell you for all of the network science metrics, but that seems to be the theme. I'll just give you one other example, which is a very common connectomics analysis to do is to look for rich clubs, look for hub-like neurons. And again, the most extreme hub-like neuron, this pair of neurons, AVEL and AVER, that one shows up as a hub in both the connectome and the signal propagation atlas. But the other list of hub neurons do not overlap. Okay. All right, so that was finding one. So finding two is that, so that was all kind of looking over here at the connectome and looking over here at the signaling measurements without integrating the two. But what happens if we try to ask a slightly different question, which is, if we have information about both, what can we learn when we combine both? And so that's what we've done here. So this is work of Matt Creamer, CV Star Fellow, with Jonathan Pillow and the SCGB. What we've done is made a connectome-constrained linear dynamical systems model. So the idea here is to take the binarized connectome, say, let's not make so much assumptions about the weights, and give it access to all of our recordings and ask it to infer a set of weights, W, and ask how well does that recapitulate our measurements. And the answer there is it actually does really well, about as well as we could expect. Here we have this dashed line showing, based on the variability in our measurements, how well we might expect our model to do. And if you constrain the model to only use connections along the wires, it can do about as well as we would expect. And really interestingly is if you just so much as permute the labels on that connectome, so you have the same sparsity, the same architecture, then the model fails. It's not able to recapitulate our measurements. And so for me, this is very strong evidence to suggest that once you give a model access to our measurements, it can do a much better job explaining how signals must flow along the wires than if you gave it some other sparse network. So the connectome is clearly important for this. So I think that is one of the strongest evidence I've found for the role of the connectome in explaining our measurements. Whoops, that's backwards. There we go. OK. But then what about this finding 3? We wanted to look at the role of extrasynaptic signaling because in the worm we have a single-cell transcriptome. We know which peptides bind to which receptors. And it turns out that if you draw a graph of the possible peptide GPCR interactions, it's extremely dense. It's a much denser network than the connectome. And so here I'm showing you that the in degree for each node is something like 40. So many, many neurons. Each neuron is listening to 40 different types of peptides and is expressing and potentially releasing many types of peptides. And some of this, a lot of this could be happening on long time scales, or could be changing excitability or acting as a neuromodulator. And that would show up as variability in our measurements. But if it's happening on fast time scales, by directly activating neurons, for example, we should see that. And so what we did is we repeated our signal propagation measurements in mutants that are defective for dense core vesicle release. And so this, among other things, stops the release of neuropeptides. And we found several, we kind of set a very conservative bar, high standard here, and found several instances, so 56 of these instances, where we see pairs of neurons that signal in the wild-type animal, but when you knock out dense core vesicles, the signaling goes away. And not only that, when you look, you see that they don't have direct monosynaptic connections and that they express the right peptide receptor pairs such that they could be communicating with one another. And so to us, this is strong evidence that, empirically, that in some cases, there's fast-ish, certainly on our 30-second time scale, peptide-mediated extrasynaptic signaling that doesn't go on the wires. And this is also consistent with our model. If you ask in general, does the model do better if you give it more wires, if you don't constrain it to the connectome? The answer is not that we can tell. But if you look at these cases where we have strong empirical evidence of extrasynaptic signaling, there the model struggles if it's constrained to only send signals along the wires. And if you allow it to add more more wires, like you might need for extrasynaptic signaling, it adds them. OK, so how do we make sense of all of this together? All right, well, I would say, what would I say? I would say that the connectome is really important for explaining our measurements, right? So that speaks to what I think Mala was excited to hear. And I would say it is also at the same time true that we struggle to predict neural responses or signaling architecture de novo. And maybe that's OK. Maybe we shouldn't expect the connectome to do that for us. And maybe having both is really valuable. And the other thing I want to say is I think understanding these extrasynaptic signaling is going to be really exciting, and it's a direction we're going. We're working with Yulong Li and Isabel Beetz and others to put grab sensors in and directly measure the spatiotemporal dynamics. I think the other really exciting direction is to try to understand where this variability in internal states are coming from. And I think I'll basically end there. But I want to thank again the Simons Foundation and take your questions. Yeah, great. Thank you, Michael. Yes to the voltage imaging. I mean, we would love to do voltage imaging. As soon as that's kind of working robustly enough, that's where we want to go. About gap junctions, and some of this may be a little complicated for me, but we do have gap junction data in the worm, and we have fairly good ones, especially from the earliest recordings. So I'm told, you talk to Mei Zhen, who's done a lot of the modern data, that some of the earliest EM data that White used, they used a different kind of freezing protocol, and dehydrated the cells a bit, and so that the cells pucker away from one another, except for where they're attached by gap junctions, and so it's very straightforward in that data set to identify those gap junctions. So we have access from that data set to separate gap junction and chemical synapse, and we've been playing around with that in some of our models. So yeah, I'd be happy to talk more about that and the status on these newer data sets. Yeah.
Okay, so I would like to understand a little bit better your measurements.
Yeah.
Let's suppose that you used, so you told us how well do they predict what the connectome would predict,
Yeah.
Or vice versa. So if you take the first 10 minutes of your 40 minutes recording,
Yeah.
and you use those to predict the last 10 minutes,
Yeah.
what do you get? And if you take one animal to predict other animal, how much correlation do you get? So can you tell us a little bit about that?
I don't know, is there a way I can mosey through my slides? Maybe not. Okay, great. I won't click all the way back, but I'll just use words. That's a good idea. Thank you. Which is, so I put up, the trained test validation across animals is, depending on your metric, it's fairly low, I would say. There is a significant amount of variability. There's different places you can see this. So if you're just looking at the average response in a certain window, it's around 16%. If you want to look at variability across animals, you can get a sense for it in a typical recording. I've sorted these by the strength of the response. And then we are doing, I don't have particularly good answers for you within and across recordings. That's something we look at. There's some technical, I think one of the things that's interesting is history dependence and looking for kind of history dependence of these responses. And one of the things that's well known is as you stimulate the neuron, even in a single recording, it tends to habituate. And so we're thinking about, there's a little bit of a play there between how often you ping the same neuron within a recording versus across. One of the things we do that I'm kind of excited about is, we can use these signaling, oh my gosh, we can use these, somewhere I have a variability slide. We can use these kernels to tell us something about variability. Okay, here. And stereotypy, and we can kind of characterize on a neuron pairwise basis where we see like bimodal responses as some of the most variable. And I think that's a really exciting place to look because that could be a substrate for neuromodulation or other factors that might explain that variability. And that's a high on our list direction where we're going. Yeah. Thanks, Pietro.
Great talk, Takaaki Komiyama here. Have you tried to fit the stimulation data with an all-to-all connectivity network?
Yes.
Can that fit the data better?
Yes. Interestingly, so I don't have my mouse, but it doesn't do much better. And it's not clear whether that's, oh, wait, maybe, no, it's hidden off here. Well, okay. It doesn't do much better, and it's not clear whether that's because it doesn't need it or because we can't see it given our trained test validation. Basically, another way of saying this, given the amount of variability we see in the animal, it's not clear whether you need those extra connections to do better than we're already doing. For the specific examples, the 56 where we see extrasynaptic signaling, it does help though, and that's one of the slides I showed.
Is the solution very similar to the connectome-constrained one?
That's a good question. That's a good
question.
How
unique it is.
That's a good question. I'd have to check. One of the things it's not similar to is it is not similar to the number of synapses you would expect. So one question you might ask is, does this model find something that matches what people often use as a proxy from the connectome for synaptic strength, which is the number of synaptic contacts. And there it has a negative correlation. But how well the unconstrained model matches the constrained model, that's an easy check. We should check that. Thank you.